This lecture covers sections C and D of Chapter 3, Vander's Physiology. We'll begin with section C, interaction between proteins and ligands. Section C picks up with section 3.8, binding site characteristics. Vanders likes to remind us that physiological processes are governed by the laws of chemistry and physics, and perhaps nowhere in physiology do we see that more prominently illustrated than in the binding of a ligand to a receptor. So we define a ligand as any molecule, or perhaps an ion, that's bound to a protein in a reversible, non-covalent fashion. So the forces that draw these molecules together, or a molecule and an ion together, can be electrical attractions or hydrophobic forces, and both of these often play a role in the interaction between a ligand and a receptor. We'll spend some time talking about binding sites in this particular chapter. And so what we'll observe is the following, that a protein may have multiple binding sites for ligands, and typically what we observe is that when a ligand binds to a binding site, we have a conformational change that occurs within the protein to which that ligand bound. We've talked previously in Vanders about the nature of proteins and the idea that they are this long chain of amino acids. It stands to reason then that if we alter the structure of one portion of this long chain, even though it's folded around into a globular shape, then that will have some impact on the structure of the remaining portion of that chain, and therefore some impact on the overall structure of the protein as a whole. And that's exactly what we see as we observe the interaction between ligands and proteins. So this conformational change typically either activates or inhibits the protein to which it binds. And we'll see examples of both of those as we move forward. Vanders also likes to remind us that structure and function are inseparably related. We certainly see that as we look at the relationships between anatomy and physiology, but here we're observing that idea at a molecular scale. We've discussed briefly the endocrine system and the binding of hormones to their receptors. And so we've talked about the importance of specificity in those bindings. The binding of a hormone to a receptor is one example of many that we observe in physiology of the binding of a ligand to a protein. So we observe that the protein binding site has a shape that's complementary to the ligand to which it binds. This enables the protein to recognize one particular ligand among hundreds, at least, that it's exposed to in its role, in its position within the body. This shape of the protein, as you're well aware, is determined by the amino acid sequence, and most frequently, this is not determined by adjacent, or at least entirely by adjacent amino acids. Our book illustrates this as we look at the picture that's located on our screen on the right-hand side. We see a protein, and most of the amino acids are shaded in orange, but we note that some of those amino acids are shaded in purple. Any amino acid shaded in purple plays a role in determining the three-dimensional structure of the binding site for the ligand. And so this clearly illustrates the idea that many different amino acids throughout the chain of a protein, not just adjacent ones, contribute to the overall structure of binding sites. We also notice that the degree of specificity for a protein to a ligand, or of a ligand to a protein, may vary widely depending on the nature of that interaction. So our book provides us with a simplified example, but a good example as we look at the diagram that's found in the middle of our screen. This is, of course, taken from Vander's Chapter 3. On the right-hand side, we see protein X. Protein X, as you'll note, has a shape that allows it to bind to ligands A, B, and C. All of those ligands bind nearly perfectly to protein X, and so it exhibits a relative low degree of specificity. On the other hand, if we look at protein Y on the right-hand side of the screen, we see that the nature of the shape of protein Y only allows it to bind to ligand C, and so it's much more specific than protein X. We see this play out again and again in the story of physiology. We talked before briefly about the estrogen receptor and how it's known as a promiscuous receptor. It binds to its intended ligand, 17-beta estradiol, but it also binds to a number of other ligands, albeit with somewhat lower affinity, as we'll talk about soon. And the binding of these unwanted, we might say, ligands also have an impact on the cell as a result of the binding to the estrogen receptor. We see another interesting example of the importance of ligand specificity in human physiology in the adrenergic receptors, receptors that bind to epinephrine. We have two broad categories of these receptors found in the human body: beta 1 receptors, which are found in the heart primarily, although not exclusively, and beta 2 receptors, which are found primarily but not exclusively in the lungs. And what we observe is that the binding of epinephrine to both of these receptor categories brings the body closer to a fight or flight response. In the case of binding of epinephrine to beta 1 receptors, we see an increase in heart rate as a result of that binding. As a result of epinephrine binding to beta 2 receptors, we see vasodilation occurring, allowing a greater amount of air and therefore oxygen to make its way into the lungs as a result of this binding. In both cases, this is adaptive because it prepares the body for a fight or flight response. However, this has an interesting story when we look at, this creates an interesting story when we look at the history of drugs that we have created to treat a disorder referred to as asthma. Asthma, of course, is an unwanted or ill-timed narrowing of the airways as a result of constriction of smooth muscle that's found in the airways associated with the human respiratory system. Originally, our treatment for asthma consisted of generic beta receptors. And so these receptors were effective in causing vasodilation of these airways. However, they also brought about rapid heart increase in the beating of the heart as a result of the fact that they bound to both beta 1 and beta 2 receptors. More modern asthma treatments are beta 2 specific, or nearly so. And so in this case, they bind to the beta 2 adrenergic receptors that are found in the airways, bringing about vasodilation and providing some relief to the vasoconstriction that is the result of asthma. However, they don't provide any appreciable increase in heart rate as a result of the fact that they don't show much affinity, as we'll talk about soon, for the beta 1 receptors that are found in the heart. We now spend some time talking about affinity, a topic that has great importance when we discuss ligand-protein interactions. Affinity describes the strength of the binding that occurs between a protein and a ligand. Essentially, it determines how likely the ligand is to leave. We've discussed affinity earlier, both in our course and in this particular lecture, in the context of, once again, for example, the estrogen receptor. The estrogen receptor binds with relatively high affinity to its intended ligand, 17-beta estradiol. It binds, hesitates there, and has an impact, triggering the effects on the cell that occur as a result of estrogen binding. However, other ligands also bind to that estrogen receptor, albeit with lower affinity. They bind, but don't have the same effect that estrogen does. Specificity is different than affinity, or affinity is different than specificity, because specificity is primarily the result of shape, whereas affinity is the result of both shape and attraction. So our book provides us with some illustrations here that will help us understand this idea. Our book provides us, as we see on the screen, with three hypothetical proteins, protein 1, 2, and 3, and one ligand that binds with different affinities to all three of these proteins. In the case of protein 1, the ligand binds with high affinity to protein 1 because the shape is both complementary, as we can see, and there is a charge, complementary charge, negative in the ligand and positive in the protein, that contribute to this binding, meaning that there's an attraction between the binding site and the ligand in this particular case. In the case of protein 2, we have an identical shape, but we lose the charge. And so this is an intermediate affinity binding site as a result of the fact that it fits just fine, but because it doesn't have that charge, it's not as likely to hesitate there to remain in that binding site for as long a period of time. In the case of protein 3, we see that we neither have the charge nor the complementary shape. And so this is a low affinity binding site for the ligand, for the same ligand, as you can see, as a result of the fact that it's a negative in both cases. So affinity has important consequences for the interaction between a ligand and a protein. For example, high affinity drugs are only required in very small quantities, and they produce very few side effects as a result of the fact that they can be used in small quantities, bind to their intended ligand, and interact with a relatively small or perhaps no other binding sites within the body. Now, it's also true that toxins that work as a ligand binding to a binding site that have a high affinity are extremely potent toxins and lethal in very small doses. We'll look at 3 examples of affinity that we observe in the natural world, not all of them tied to human physiology, but all of them prominent examples in biology of the importance of affinity in ligand-protein binding. Number 1, carbon monoxide binds to hemoglobin with an affinity that's 210 times greater than the affinity with which oxygen binds. This is exceptionally important and an example of a situation where a toxin that binds with very high affinity can be extremely lethal. This is what makes carbon monoxide so dangerous. It binds to hemoglobin and remains bound, and if there is a sufficient amount of carbon monoxide that's bound to the hemoglobin, then the hemoglobin cannot pick up an adequate amount of oxygen to deliver throughout the body, and the body dies from lack of oxygen, despite the fact that there's plenty of oxygen available to it. It's just that it's unable to access that as a result of the fact that a higher affinity ligand is bound to oxygen's binding sites. So we're aware of the fact that this creates a very dangerous situation in any scenario in which a combustion engine is run inside, and so a car in a garage, for example, a electric generator that's run right outside a window can have lethal effects on the individuals inside as a result of the fact that carbon monoxide has such a high affinity for hemoglobin. We also notice this, that when we talk about thyroid hormone, we'll see that thyroid hormone is produced produced in one form, but turned into another form within the body. So T4 is known as thyroxine, and T3 is known as triiodothyronine. We notice that thyroid hormone is produced as T4, thyroxine, but later converted to T3 by enzymes that are found within the cells where the thyroid hormone receptors are located. And so T3 is really the biologically active form of thyroid hormone, but T4 is the form in which thyroid hormone is produced. The adaptive feature of this is that it provides a reservoir of thyroid hormone in the body, and the body is able to convert that T4 into T3 as it's needed in these cells. Lastly, an example from outside of human physiology, ribisco and PEP carboxylase. You're aware of the fact that these 2 enzymes are responsible for carbon fixation, for taking CO2 from the atmosphere and turning it into sugars during the process of photosynthesis in plants. Ribisco, in fact, is an enzyme that's unique in that it's the most abundant enzyme that's found on the planet, the most abundant protein found on the planet as well, by the way. But interestingly, ribisco has a very low affinity, or at least a relatively low affinity for carbon dioxide. And so PEP carboxylase is an enzyme that's found in C4 plants and allows these C4 plants to close their stomata and continue to conduct photosynthesis, which allows the C4 plants to live in dry conditions. And so this story of affinity, the strength of the bond between a ligand and a protein, is important in numerous aspects of biology. We also discussed saturation in our discussion of the interaction between proteins and ligands. Saturation describes the fraction of binding sites on proteins that are occupied at any particular point in time, and it depends primarily on 2 factors. Number 1, the concentration of the unbound ligand. We see this illustrated by our text in the upper graph that we observe on the right-hand side of this particular screen. We have ligand concentration found on the x-axis of this particular graph, and we have percent saturation found on the y-axis. We have a nice little illustration up here at the top that shows us the ligand shown in green and the proteins shown in brown, a light brown, maybe tan, and you can see the binding site for the ligand on the proteins. So when we start out with the low ligand concentration, concentration A, shown on the x-axis on this top graph, we can link that to the first square that we see here. We have a low concentration of the ligand. We have a handful of protein molecules that are present here. That concentration of protein molecules doesn't change as we go throughout the course of this little hypothetical experiment. But we see that we have one of the four protein binding sites that are occupied, so 25% saturation at a ligand concentration of A. If we go to B, we see that we have increased the ligand concentration as we move from left to right on the x-axis. We're increasing the ligand concentration. We see, if we look at the y-axis, that we have moved up to a 50% saturation state on the graph. And if we look at our little diagram, our box there, then we see that two of the four proteins have ligands bound to their active site, and so, of course, that gives us the 50%. If we go over to C, we see that we've increased the ligand concentration further. And we see in this case that we've moved up to a 75% saturation state. We see that corresponds to the little diagram that we have at the top. Interestingly, at D, we reach an important point. We see that we have reached 100% saturation. We've increased the ligand concentration. And now, if we look at box D, we see that each binding site on all of the four proteins is bound to a ligand. Furthermore, as we look at the remainder of the x-axis, we see that we continue to increase the ligand concentration, but we see no change in the percent saturation because we're already maxed out. You can see very clearly that there are no more proteins with open binding sites to allow the ligand to bind. So clearly the concentration of unbound ligand has an impact on the percent saturation. If we look at number 2, we see that affinity, as you would imagine, also has an important effect on the saturation state of a protein. So interestingly, as you would imagine, higher affinity proteins, or proteins that possess higher affinity in their active sites, require less ligand in order to reach a relatively higher saturated state, and eventually a less ligand is required to reach saturation. We can see that in the second diagram that's shown for us on this particular slide. We have two proteins, protein Y, which is shown in blue here, and at the top in our little square, which has a high affinity binding site, and protein X, shown in purple at the top, which has a low affinity binding site. And so, as we move across the x-axis here, we're adjusting the concentration of the ligand, and the ligand again binds to both of these proteins. And so what we can see is this. When we reach this particular point, which was equivalent to about a 50% saturation state in the case of our first experiment, then we notice that for the protein X, which is low affinity, then we are only 25% saturated. But if we look at protein Y, shown by the blue line here, then at the same ligand concentration, we have achieved 50% saturation. And so that's what we're looking at up here in these little boxes. You can see that the purple protein with the low affinity binding site, only one of four active sites are bound at this particular ligand concentration, whereas the protein Y, which has the high affinity binding site, has two of the four active sites bound at this particular protein concentration. It stands to reason that because the ligands bind to and stay longer at a high affinity binding site, that it would take less time to reach saturation with their binding sites relative to the binding sites that we find on a low affinity, a protein that possesses low affinity binding sites. We also note that competition can often play a major role in the interaction between ligands and proteins. Competition occurs when two ligands compete for the same binding site, a process referred to as competitive inhibition. In competitive inhibition, one ligand acts as an antagonist as opposed to the native ligand, the ligand that typically binds in normal physiology to that binding site, which acts as the agonist. The agonist binds and has an effect, but the antagonist binds and has no physiological effect. We see various examples of this in physiology, oftentimes in a situation in which we are providing treatment for some type of pathophysiology. Naloxone is an example of competitive inhibition. It's a high affinity molecule that binds to opioid receptors in the central nervous system. And so we see an example of the effects of naloxone, or actually any competitive inhibition situation, in the little graph shown in the inset on the right-hand side of the page. We have the activity that's brought about by the binding of the agonist on the y-axis. We have the concentration of the antagonist that's found on the x-axis. And then we have the concentration of the agonist, which remains unchanged throughout this experiment, shown at the top of our little graph there. So what we observe is the following. As we increase the concentration of the agonist, we see a corresponding decrease in the activity that's brought about by the agonist, simply because of competitive inhibition. The concentration of the agonist doesn't change, but the concentration of the antagonist progressively increases in this case as we go from left to right across the x-axis. And so naloxone has been a life-saving measure that prevents death from overdoses as a result of the fact that it binds to those same receptors that illegal drugs, heroin in particular, are binding to. A good deal of human physiology revolves around this idea of regulating protein activity. And so here we talk about two methods in human physiology that are utilized to regulate protein activity. Number one, changing the shape of a protein, and number two, regulating the synthesis and degradation of a protein. At this particular point in time, we focus on changing protein shape and the methods by which that is accomplished in human physiology, or any vertebrate animal physiology. Regulation of synthesis and degradation of proteins is commonly used to regulate protein activity within the body. Many proteins are what are referred to as inducible proteins. These include a number of our hepatic proteins. These proteins increase in synthesis. The synthesis of these proteins increase, and therefore the concentration of these proteins increase following the contact of the body with the appropriate ligand. For example, if you eat a burnt marshmallow, you're taking in those products of combustion, which are mildly toxic, and those products trigger the synthesis of hepatic enzymes, such as cytochrome P450-1A. These enzymes are produced in higher levels in response to the ligand to which they bind. Again, we spend most of our time here talking about changes in protein shape, and we'll begin now by talking about allosteric modulation. And so allosteric modulation involves the binding of a ligand that alters the shape of the binding site to typically a different ligand, but not always. What we observe is a nice little diagram from our text that shows us, that illustrates this particular process. And so in this particular protein, we have both a regulatory site shown on the bottom to the left-hand side and a functional site. If this was an enzyme, we would refer to this as an active site that's shown on the top of the protein. And so the regulatory site binds to a regulator molecule or a modulator molecule, shown in purple here. And when it binds, it changes the shape of the functional site, either allowing or not allowing the ligand to bind to the functional site. And so in this particular manner, the protein can be turned on and off depending on the presence of the modulator molecule. We also mention, in the context of allosteric modulation, the idea of cooperativity. Cooperativity involves multimeric proteins, proteins that are involved of multiple protein chains held together by non-covalent bonds. This, of course, is the quaternary or fourth level of protein structure that was discussed earlier in our class. In this particular case, what happens is the following. When a ligand binds to one of these chains, it changes the shape of that chain, which influences the shape of the other chains that are part of the protein, and therefore influences the likelihood of those chains to bind to their intended ligand. A good example of this is hemoglobin. Hemoglobin each chain of hemoglobin has an increasing affinity for oxygen depending on if the other chains that are found within the four chains that make up hemoglobin are bound to oxygen or not. In other words, if one of the chains is bound to oxygen, then the three remaining chains have a higher affinity for oxygen. Lastly, in the context of protein regulation, we'll talk about covalent modulation. This is another method of altering the characteristics of a functional site, known as an active site in the case of an enzyme, and therefore regulating the activity of a protein. Almost always, when we're talking about covalent modulation, we mean phosphorylation. Phosphorylation occurs when an enzyme known as a protein kinase transfers a phosphate, the third phosphate, from an ATP to a protein. This represents a transfer of energy and changes the shape and therefore the activity of the receiving protein. This is referred to, of course, as phosphorylation, and the protein remains activated. In other words, the shape of the protein remains altered as long as that phosphate is in place. A phosphoprotein phosphatase is responsible for dephosphorylation. It removes that added phosphate and allows the protein to return to its original shape. We see that occurring in the diagram in the lower portion of our slide. We see a non-phosphorylated protein on the left-hand side. We see a protein kinase adding a phosphate. We see a change in the functional site as a result of that addition that enables the ligand to bind to the functional site. And then we see a phosphoprotein phosphatase removing that phosphate and allowing the protein to return to its original conformation. This is a very common method of protein regulation within physiology. In section D of chapter 3, we get to talk about enzymes and chemical energy. In section 3.10, we talk about metabolism. Metabolism is collectively the thousands of chemical reactions that occur per second within a cell and result in both the overall production of molecules within a cell and the overall consumption of ATP by that cell. There are both anabolic reactions that occur within the cell. These are reactions that are putting portions of a molecule together to create new molecules, and there are catabolic reactions that occur within the cell, reactions that are breaking molecules down. In each of these reactions, energy is either added or released. This energy is measured in the form of kilocalories, and when we look at the overall reaction rate of these reactions, we can measure the speed of these reactions by the change in either the reactions or the reactants or the products per unit time. As we'll talk about in just a moment when we conduct enzymatic reactions in vitro. So the reaction rate is most frequently impacted by four different factors. Number one, reactant concentration. Number two, the activation energy of the reaction, which is specific to the molecules involved and involves the repulsion of the electrons that are involved in the particular reactions, the temperature at which the reaction is occurring, and lastly, whether or not a catalyst, in our case, typically a biological catalyst known as an enzyme, is present to speed up the reaction. So, interestingly, what we see is that the easiest way to measure the progress of these reactions is to measure either the, as we said, degradation of a reactant or the production of a product. One such reaction is the reaction that's catalyzed by the enzyme cytochrome P450-1A. This particular enzyme is a hepatic enzyme that we mentioned earlier in this particular lecture that's responsible for the detoxification of xenobiotics that make their way into the diet via food. And so a xenobiotic is a term for any molecule that is not normally found within the body. We often think of in vitro as meaning in a test tube. In reality, in vitro means a reaction or an experiment that's conducted in a laboratory setting outside of a living organism. So in vitro, cytochrome P451A, also known as P451A, converts ethoxyresorufin, an orange non-fluorescent substrate, to resorufin, a pink fluorescent substrate. This is a convenient way of measuring the speed of this chemical reaction, this enzymatically catalyzed chemical reaction, by measuring the production of a product per unit time. Resorufin fluoresces can be measured with a plate reader commonly used in protein studies and molecular biology, and therefore the progress of a cytochrome P451A reaction and the number of P451A molecules present can be quantified with great precision. The vast majority of chemical reactions that occur within the body are catalyzed by molecules referred to as enzymes. Enzymes are nearly always proteins, and enzymes are responsible for catalyzing these reactions because most of these reactions possess relatively high activation energies. This means that there are two means by which we could cause these reactions to occur. Number one, we could increase the temperature, therefore meeting the requirement for sufficient activation energy. But the problem is that doesn't work out well in cells because an increase in temperature leads to protein denaturation. So number two, in living systems, most frequently this activation energy requirement is met through the provision of a biological catalyst known as an enzyme. Enzymes are responsible for reducing the activation energy. They catalyze both the forward and reverse reactions, and they reduce the activation energy by providing a site known as an active site. This is the binding site that we discussed previously in our discussion of protein-ligand interactions. The term active site is simply a term that's used exclusively to refer to that binding site when it occurs on an enzyme in the context of an enzyme-ligand reaction. And of course, you realize that in the case of an enzyme-ligand reaction, the ligand is referred to as a substrate. Enzymes are most frequently named, or at least frequently named, based on what they do. Alcohol dehydrogenase, for example, is responsible for breaking down alcohol. Another example of a hepatic enzyme. DNA polymerase is responsible for synthesizing the polymer of DNA, synthesizing DNA, which is a biological molecule that's a polymer of nucleotides. We also noticed this, that enzymes work very, very quickly. Our best estimates of the rate at which enzymes work are in the neighborhood of 2,000 times per second. Yes, we're stating that a substrate binds to the active site of an enzyme, is turned into a product, released, and then another substrate comes and binds. And this process occurs 2,000 times in a single second. These molecules, of course, are very small and moving very rapidly, which explains how we can have such high enzymatic rates. We also notice this, that of course enzymes are no exception to the protein-ligand interactions that we talked about earlier. Shape is very important in terms of the interaction with an enzyme and its substrate, and enzymes only bind to certain substrates. However, we do also notice that when a substrate binds to the active site of an enzyme, we do see a slight change in the shape of the active site of the enzyme as the result of that binding. So this is sometimes confusing as we start to think about this particular idea because these two statements seem to be telling us contradictory things. Interestingly, though, our best data indicates that both of these are simultaneously true. The lock and key model, shown on the bottom left-hand side of our slide, supports the idea that substrates only bind to enzymes with an active site that has a complementary shape and allows them to bind. However, on the right-hand side, the diagram that we see shows us the induced fit model. The induced fit model says that substrates bind to an active site and change ever so slightly the shape of that active site in order to bring about a sufficient binding or an appropriate fit of the substrate to the active site. We also turn our attention briefly to the diagrams that are found at the top of our slide before we move on. The top graph, two graphs shown in blue and green there, show us the effect that the addition of enzymes have on the overall number of reactant molecules that can participate in the reaction. On the y-axis of each graph, we have the number of reactant molecules present, and on the x-axis, we have the energy of these reactants. And so you can see that the majority of the reactant molecules have an intermediate amount of energy. A smaller number have a lot of energy, and a smaller number have a low amount of energy. So in the absence of an enzyme, which is what's shown on the blue and green top graph on the left-hand side, the one I'm circling now, we see that in the absence of an enzyme, the activation energy is relatively high, and a relatively small number of molecules have sufficient energy to undergo the reaction in the absence of an enzyme. However, what an enzyme does is greatly lower that activation energy. And so if we lower the activation energy, we see that in this second graph, approximately half of the molecules that are shown in the graph have sufficient energy to undergo this particular reaction. The two graphs on the bottom of our figure in the upper right-hand portion of the screen show us another illustration of this idea of activation energy. This particular graph that I'm circling now shows us the energy on the y-axis and the progression of the reaction on the x-axis. And so the reactant starts out with more energy than it finishes with. This is an exergonic reaction, a reaction that gives off energy. But the red bar there, or the red wall, indicates or signifies the activation energy that's necessary for the reaction to occur. If the reactant can make its way over the wall, then it will proceed to completion of the reaction. However, and eventually release energy. However, some initial energy investment is required in order for the reaction to be kicked off or to initiate. On the right-hand side, the graph that we see shows us the activation energy in the presence of an enzyme. We see that the activation energy is significantly less, making it much more likely that that reactant will have sufficient energy to make its way over the activation energy and proceed to completion. In both cases, the reaction releases the same amount of energy. The overall energy change is exactly the same. It's just that in the presence of an enzyme, we have lower activation energy, so it's more likely that those molecules will be able to undergo the reaction. We might also mention this. We could ask the question, how does an enzyme lower the activation energy of a reaction? And the answer lies in the shape of the active site or the nature of the active site. The active site provides a microenvironment that allows the reaction to occur. There are different possibilities for the composition of this microenvironment. One is that as the substrate binds to the active site, it bends and twists the bonds of the substrate ever so slightly, putting stress on certain bonds and making it much more likely that those bonds will break, producing the products. Another possibility is that as the substrate binds to the active site, it provides an environment of a slightly different pH that makes it more likely, once again, that bonds will either break or form in that microenvironment of the active site. We also notice that many enzymes require cofactors and are unable to catalyze reactions without these cofactors. Cofactors are typically required in very small amounts. They bind to the active site of an enzyme, such as the diagram that's shown on our screen. So if we look at the lower right-hand side of the screen, we see the enzyme, and the amino acids of the enzyme are shown in blue, but the cofactor is shown in red. The cofactor binds to a small portion of the active site of the enzyme and allows the substrate to bind and the substrate to catalyze the reaction. Cofactors that are organic molecules are referred to as coenzymes, and NAD+ and FAD are good examples of coenzymes that are frequently encountered in cell physiology. Coenzymes often participate in reactions by receiving or donating hydrogen ions, as is depicted on the screen. A good example of the importance of cofactors in human health is the pathophysiology, or the disorder scurvy, depicted in the lower left-hand corner of our screen. Scurvy was a disorder that was commonly encountered by sailors who would be gone on long voyages and become deficient in vitamin C. Vitamin C serves as a cofactor for enzymes that are responsible for the synthesis of collagen in the human body. Collagen is a large protein that plays an important role in skin and bones and ligaments and blood vessels in the body. When the body is unable to synthesize collagen, these portions of the body weaken. In the case of scurvy, symptoms are encountered and death often ensues as a result of bleeding that comes from the failure of the body to maintain the integrity of blood vessels. We now spend some time talking about how the cell regulates enzyme-mediated reactions. There are multiple mechanisms that are utilized to regulate the speed of enzyme-mediated reactions, one of which is regulating the substrate concentration. If this is depicted on the top graph on our screen, and if we look at the top graph on the right-hand corner of the screen, we see reaction rate on the y-axis and substrate concentration on the x-axis. We see as substrate concentration increases, reaction rate increases until we reach saturation. And so, of course, when we reach saturation, every single active site is bound to a substrate at every possible moment. So the addition of more substrate has no impact on the overall reaction rate. Another possibility for regulating enzyme-mediated reactions is adjusting the enzyme concentration itself. We see this depicted in the second graph shown on our screen. We have one enzyme at two different concentrations. We see the orange line indicates the enzyme concentration at half the green concentration, and we see that the saturation rate, the reaction rate at saturation rather, is about half what we see at 2 times the enzyme concentration. And so, therefore, simply we can adjust the number of enzymes that are present and adjust the rate and therefore the number of product molecules that are produced by an enzymatic reaction at any given time. Many enzymes in the body are what we refer to as inducible. These are enzymes in which the substrate concentration can be modified dramatically over a relatively short period of time when the body needs to adjust the amount of product that it's receiving from a particular enzyme-mediated reaction. The last possibility is that we can adjust the actual activity of the enzyme. And so in this case, we're adjusting the properties of the enzyme using allosteric or covalent modulation, as we discussed previously. And if we are below saturation, this is going to have an impact on the overall reaction rate of the enzymatically catalyzed reaction. Here, we see this depicted in the lower right-hand corner of our screen. The orange line indicates the initial affinity of the enzyme for the substrate, and so we can see the progression of the reaction as we increase the substrate concentration. The green line indicates when we have adjusted the shape of the enzyme to increase the affinity. We can see in this case that we greatly increase the binding of the ligand to the protein or the substrate to the enzyme. And so we're approximately doubling the reaction rate in an identical substrate concentration, if we look at the black line there, close to the left-hand side of the graph. And this illustrates the method by which we can adjust the properties of the enzyme and thereby adjust the products that are being produced per unit time from that enzyme-mediated reaction. We also note at this point that multiple of the methods that we just talked about can be used simultaneously to adjust the rate of an enzyme-mediated reaction. For example, on this particular slide, we can see that we have 6 sites total. 1 site of covalent activation, 2 sites of allosteric activation, 1 site of allosteric inhibition, actually 2 sites of allosteric inhibition, and 1 site of covalent inhibition. And so there are binding sites for 6 different ligands there, and depending on which binding sites, allosteric binding sites, are occupied at any given time, we would expect to see drastic changes in the shape of the active site and therefore the ability of the active site to interact with the ligand. This allows the cell a great deal of flexibility. It's just not turning the enzyme off or turning it on, but it can adjust the rate at which an enzyme is working at any given time to meet the needs that the cell has for a particular product at a particular point in time. Our discussion up to this point has been leading to the topic of multi-enzyme reactions. These are referred to as metabolic pathways, and these are situations in which a number of enzymes are placed together to produce a particular product. Let's look at the upper right-hand side of our screen. In this particular case, we see four enzymes, enzyme 1, 2, 3, and 4, and enzyme 1 takes substrate A, turns it into product B. Enzyme number 2 takes this B, which was the product of enzyme number 1, and utilizes it as a substrate to create product C, and so forth down the line until we reach the final product, product E. This is referred to, as we mentioned, as a metabolic pathway, and these are universally used in the body to produce products that are needed by the cell. We also have multiple opportunities to control these metabolic pathways based on the mechanisms that we've discussed over the past couple of slides. On this particular slide, we'll first talk about end product inhibition as a mechanism of controlling metabolic pathways. In this case, we note that E, the end product, acts as an allosteric modulator of enzyme number 2, binding to enzyme number 2 and slowing down the activity of enzyme number 2. This is a form of negative feedback. So in other words, as long as there's plenty of product E in the cell, E is going to act as an allosteric inhibitor of enzyme number 2, slow down the overall metabolic pathway, and the cell won't waste energy producing E when it already has E. However, when E goes away, E will no longer be able to exert that negative feedback via the allosteric binding to enzyme number 2. The pathway will fire up again, and E will once again be produced. It's an adaptive form of negative feedback that we observe at the cellular level in almost universally in cells of the body. But we can utilize the techniques that we've talked about over the past few slides in other ways to control metabolic pathways. In the middle of our slide, we see an example in which we're controlling the direction of a metabolic pathway by turning off or on a particular enzyme. For example, in this particular pathway, if we turn on or promote the activity of enzyme number 4, we're promoting the production of E, product E, ultimately from substrate A, through the activity of enzyme 1, 2, 3, and 4. However, if we inhibit enzyme 4 and activate enzyme number 5, enzyme number 5 converts X to Y, but at the same time it converts E to D, and therefore provides the substrate for E3. And so ultimately, by turning on enzyme number 5, then we're going from product or substrate, as it is in this case, E, to product A. So the direction of a metabolic pathway can be controlled by the activation or inactivation of a pair of enzymes. Lastly, we see that oftentimes, almost universally, to be honest, in metabolic pathways, we have branches that occur. And so in this particular case, product C can go and be turned into product D product C, it's the product of enzyme number 2, but it's the substrate for enzyme 3 and 6. And so C can be turned into D by enzyme 3, or C can be turned into F by enzyme 6. So if we activate enzyme 3, then we produce more of E, because D is then turned into E by enzyme number 4. But if we activate enzyme number 6, then we create more of G, because F is turned into G by enzyme number 7. So we can modify the direction of a metabolic pathway by modulating the activity of enzymes at these branch points along the pathways. Interestingly, we see that these branched metabolic pathways play a very important role in human physiology. And unfortunately, there are a couple examples of pathophysiology that illustrate this quite prominently. If we look at the metabolic pathway on the screen, we see two enzymes, and if these enzymes are mutated, then we have distinct physiological disorders associated with these mutations. Enzyme number 6 is responsible directly for the production of melanin. The absence of melanin leads to a condition referred to as albinism. The snake you see on the lower right-hand side of the screen is suffering from albinism. And so the albinism is a direct result of a mutation in the enzyme that produces melanin. If we back up in the pathway just a bit to enzyme number 1, we see that enzyme number 1 converts phenylalanine into tyrosine. The absence of enzyme number 1, or a mutated enzyme number 1, means that all the phenylalanine that's available is a substrate, serves as a substrate for enzyme number 2 and is turned into phenylpyruvic acid. Interestingly, a mutation in enzyme number 1 is a disorder known as phenylketonuria, abbreviated PKU. This is one of several metabolic disorders that are tested for at birth when a human is born, and these are referred to as inborn errors of metabolism. The reason that we test for it will be clear if we explain the symptoms and the treatment of this disorder. The symptoms are as follows. If it's untreated, then phenylpyruvic acid builds up at very high levels, and the child develops with severe cognitive disorders. Typically, this is characterized as mental retardation, and this is an irreversible state if phenylpyruvic acid builds up and remains at this high level during childhood. However, the treatment is relatively simple, although difficult to implement. The treatment is simply dietary, regulating the amount of phenylalanine in the diet. If the amount of phenylalanine in the diet is kept minimal, then the child develops completely normally. In fact, people who have phenylketonuria must regulate very tightly the amount of phenylalanine that's found in their diet throughout the course of their lifetime. We see this reflected in common food items that are available in grocery stores. This is a Diet Mountain Dew. If we look closely at the label on Diet Mountain Dew, at the very bottom, we see phenylketonurics, contains phenylalanine. This is a warning for people who suffer from phenylketonuria that this product contains phenylalanine, and it's a product they should use sparingly.